Are you looking to quickly get an audience excited about your research? Do you want to explain your research for your family but aren't quite sure how? A lightning talk or chalk talk might be the answer for you. I'm Madison Edwards, and today I will be sharing some advice on how to construct a 3-minute chalk talk. So first of all, what is a chalk talk or a lightning talk? I will use those terms interchangeably. I would describe this as a brief 2 to 3 minute discussion of your research and its significance, and in which you will draw pictures or use images to engage your audience. So what should we consider before getting started? First and foremost, who is your audience? Is it people in your specific field? So for example, chemists researching cancer therapeutics, so a very specific topic. If this is the case, your chalk talk can be more detailed, use field-specific terms, and you may not need to spend as much time explaining the background or significance. On the other hand, if your audience is people in your general field, such as other chemists or scientists, but not in your specific subfield, you might be able to assume they have a certain level of knowledge, but you may need to define more field-specific terms and explain the significance of your project more. Finally, is your audience the general public or non-scientists? In this case, you may need to keep it more general and increase the background section and introduction, and really try to avoid field-specific terms, which we often call jargon. Second, consider how much time do you have. This will help you determine how much time to spend on each section of your talk. Let's dive into the structure of a lightning talk. Here I'll give you some advice for each section. I always like to tell people to watch a short TED Talk. I think this is a great way to kind of get an idea of how to structure your talk. My advice would be to start your chalk talk off with a takeaway sentence, or to tell your audience what you're about to tell them. I think this will help your audience stay more focused, and since they'll have an idea of your conclusion, they can have an easier time following the main points. Ideally, this takeaway sentence is the one idea that you would like your audience to remember, and it summarizes your research goal and how you will or have addressed it. This portion of your talk should only take about 20 seconds. For the next minute to a minute and a half, you can address the background of your project. Questions to consider answering are, why should your audience care about this topic? Keep in mind this will need to be tailored to the audience you're presenting for. What is known in your field and what needs further investigation? What are you going to investigate? Next, you can move on to the question and hypothesis for the next minute to minute and a half. What are your goals? What methods will you or have you used? How are you going to address your question? Consider listing out your goals or drawing an image here for your audience to reference. Again, be mindful of who your audience is when determining how detailed to get and what terminology you want to use. Finally, wrap up your presentation by returning to your takeaway sentence or summarizing your chalk talk backwards. Here, you want to reinforce the main points you hope the audience will remember. I would also like to give you some general tips for a chalk talk. As I mentioned before, avoid jargon or very, very technical terms where possible. Have a clear and logical flow to help keep your audience's attention. Practice a lot and gain feedback from people who are representative of your audience. I would suggest incorporating images and hand gestures into your work, as this will help engage your audience. Move around, gesticulate, vary the tone of your voice, emphasize certain statements, smile, show excitement, etc. These are all ways to keep your audience engaged throughout your talk. Spend some good time on your takeaway sentence so that it succinctly encompasses everything you want your audience to remember. And finally, watch some TED Talks to get ideas of how to engage an audience and create an effective takeaway statement. Let's see if I can incorporate my advice into a chalk talk. Today, I'm going to describe why protein synthesis regulation is critical to our development and how I hope to learn more about this process in order to understand disorders such as Fragile X syndrome. Many of you are probably familiar with the central dogma of biology, which describes the flow of genetic information from DNA to RNA into proteins. In this process, which is occurring constantly in our cells, DNA is transcribed into RNA, which is then translated into proteins, which are the cellular workhorses responsible for carrying out cellular functions. During this process, organisms rely on the accurate and regulated translation of mRNA by a cellular machine called the ribosome. It is essential that the mRNA is read and converted into the correct protein sequence, and that the mRNA is converted at the appropriate levels based on the needs of the specific cell type. If this process is disrupted in any way, it can be detrimental to the organism. Since translation is such a crucial process, there are proteins whose function it is to regulate the production of other proteins. Fragile X mental retardation protein is one such protein. When this protein is not produced, the process of decoding mRNA to produce proteins is disrupted, causing Fragile X syndrome. This is a worthwhile avenue to study, as in humans, Fragile X syndrome is the most prevalent form of inherited mental retardation and the primary monogenetic cause of autism spectrum disorders, which are currently on the rise. In order to investigate the role of Fragile X mental retardation protein in regulated translation, my first plan is to determine which mRNAs my proteins interacts with. Does it bind to them tightly, or does it not bind to them at all? I can then use in vitro translation assays to determine which mRNAs my protein regulates the translation of, and determine whether this leads to a decrease or an increase in production of certain mRNAs into the proteins, or whether there is no real effect on the translation of this mRNA into proteins. Finally, I hope to have an idea of whether my protein regulates all these mRNAs in the same manner, or if it prefers to regulate mRNAs that it interacts with more tightly. By looking more closely at the mechanism through which this protein regulates translation, I hope to provide researchers with information that can be used to design therapies or help create treatments to prevent or treat disorders such as Fragile X syndrome. I hope you all found this helpful and that it will assist you in writing a chalk talk about your own research.